I believe that brain aging is a program that leads to neurodegenerative diseases that we can reprogram using drugs. And before I tell you about how we plan to do that, I want to tell you why I'm doing this. So these are my grandparents. And so when I was a little kid, my grandmother suffered with Alzheimer's, and my grandfather took care of her in the home. And this had a very big impact on me as a kid, led me to dedicate my life to coming up with better therapies for Alzheimer's disease. And now, unfortunately, it's in the next generation. My aunt is currently suffering. This is a very common story. So there are more than 35 million people in the US that have an age-related neurodegenerative condition, including Alzheimer's and Parkinson's and related dementias. A little bit of background on what these diseases are. So there's a very simple problem in most neurodegenerative diseases, which is death of neurons. And depending on where those neurons die, that's the disease that you end up with. So if the neurons die in the hippocampus, that's Alzheimer's. If they occur in frontotemporal lobes, that's frontotemporal dementia. In the spinal cord, that's ALS. So the big question has been, why do the neurons die? And this is really important in order to design therapies to prevent those cells from dying. So there's been a hypothesis, at least in Alzheimer's disease, for the last 25 years called the amyloid hypothesis. It's focused on toxic buildup of proteins in or around neurons. And this is very controversial. So whether or not this is the cause of neuron death is still unclear. It's definitely not the entire picture. There's been publications that have been retracted. There's counter evidence. So there's people that are walking around with brains full of these toxic proteins that have completely normal memories. So that kind of goes against what you would think of something that's causal, counter evidence. And so for the last 25 years, there's been billions and billions of dollars spent on clinical trials that have all been aimed at one thing, which is removing these proteins, with the idea that if you get rid of the proteins, then you can cure dementia, and that will end up treating Alzheimer's disease. So there's been a couple of success stories just this year that have followed decades of failure. So there's been 2 approved drugs that have taken this strategy. So they're antibodies, they remove these amyloid plaques. The drugs work a little bit. They have been approved by the FDA. They don't work for everyone. They work better very earlier on in the disease, and they have bad side effects. So 20% of people will have brain swelling and bleeding. And so patients are now really struggling with whether these drugs are worth taking. So I believe that removing amyloid is like trying to protect the trees in a forest fire by sweeping up the ashes afterwards. It's not going to bring back the trees. And we need a preventative strategy that prevents neuron death in the first place. So we know that brain aging is the largest dementia risk factor. There's an exponential increase in risk. You may even think of it as inevitable or part of the aging process, given that by the time we're 95, 50-50 chance of having Alzheimer's disease, which are just huge odds. And I think a lot of us will be facing that with people living longer and better therapies for other diseases. So what is happening during brain aging that may be causing this? So a few things about brain aging. So some forms of cognition are relatively stable throughout your life. So wisdom, so occupational and world knowledge, remains relatively flat or unchanged, but other forms of cognition change steadily, even starting in your 20s. So working in long-term memory and processing speed. But importantly, people have different rates of this change, and some people don't decline hardly at all. So these are just longitudinal graphs of people at 2 different time points, and each graph is a different aspect of cognition. So if you look at word retention, for instance, some people measured at 2 different time points have a completely flat line, so they don't lose this ability at all, measured over decades. But some people have this relatively steep decline. And the reason this is important is it offers some hope that not everyone is having this age difference. And the other thing that happens during brain aging is that brains actually shrink. This also starts in your 20s. So these is an average of MRI gray matter volume over time, and you can see on average people's brains are shrinking. That again is variable, so some people have large amount of loss, some people have hardly any loss. Underlying that are what's happening within cells. So at least during normal aging, neurons aren't dying for the most part. Instead, the shrinkage is due to loss of dendritic processes and synapses. And underlying, and there's other hallmarks of aging. And I should say that these hallmarks in the brain are specific, they're different. So yesterday, Steve Horvath was talking about splitters versus lumpers. This is a little bit of a splitters view, which is that there are some very specific hallmarks that are occurring within brain cells that you may not see otherwise because you, for the most part, have the same neurons you've had your entire life. So mechanisms like telomeres and senescence are less important. Instead, you see mitochondrial dysfunction, losses of dendrites, changes in neurotransmitter signaling, and then in glia, you have this glial dystrophic phenotype. And their gene expression changes that happen with age mirror these hallmarks. So about 10% of the genome changes with age in human prefrontal cortex. You have cells. Inside the cells, you have your nucleus, you have DNA. The DNA is transcribed into RNA, which is translated into protein, and those mRNA levels change with age. So here's a couple of examples. I think they're very striking. These are not subtle changes, and they, again, like cognition, like changes in gray matter volume, are starting in your 20s, so this continuous process. So these are 2 disease-related genes. You see that from age 20 all the way up to age 80, you have these decrease in this gene calpanin. These are both Alzheimer's-related genes, and it's pushing in the direction that would be predicted to cause a disease. And so the question is, can you take these levels that are disease-causing and turn them back to levels that are happening in your 20s? And how would you do that? So you can take all of these, turn it into a clock. This is a transcriptome clock in human brain. And what we showed in large longitudinal cohorts of older adults that were followed before they died and then donated their bodies to science, that people that are just 5 years younger in this transcriptome clock are 6 times less likely to have dementia. They're also less likely to have preclinical Parkinson's signs, and they're in subjects without any disease at all. They have slower changes in their rate of cognitive decline. And for purists, delta age in human brain also predicts overall lifespan, if that's your bottom line, and that only happens in people older than 60, which makes sense because people under 60 are not dying from neurodegenerative disorders for the most part. Okay, so some people, these people that have slower brain aging, these are the people that are 95 years old, they're sharp as a tack, have genes that is related to how fast their brains are aging. And we can determine which genes are controlling the rate of their brain aging using causal inference and Mendelian randomization. And so what NeuroH does is creates drugs that target these proteins, we're calling them BOS proteins, that then can reverse the program of brain aging at a transcriptome level. And if Yamanaka factors are the CEOs of the cell, these are more like middle management. So they're not necessarily transcription factors, but they do regulate gene expression. So NeuroAge has two products. Our first product is a test that can tell you how old your brain is and your likelihood of having dementia more than 30 years ahead of time. And this test also is continuously feeding data into our personalized therapeutics. So this is Max. Max is 55. He has a brain age of 51, so he's 41% less likely to have dementia, and that's pushed further out. So currently we can recommend to Max to improve his lifestyle and come back and get retested. And then coming soon are NeuroAge's personalized therapeutics for people that aren't able to achieve low risk of dementia with lifestyle interventions alone. And so the NeuroAge test is pretty comprehensive. It has a blood test for our RNA-seq levels, genetics, epigenetics, there's a brain MRI, and there's also cognitive testing. And we put that all together into a super predictor using AI, and we can tell you how old your brain is. And then just a little bit more about our drugs targets. So we're pretty excited about these. These are the BOS proteins that we're targeting. Many of them already have evidence in mice, are linked to neurodegenerative diseases. They're in logical pathways. There's an overrepresentation of mitochondria, so I'm a big believer that mitochondria are driving a lot of this. Many of them also have links to neurodegenerative diseases, and in some cases we can move quickly because they're repurposable or modifiable drugs that exist for them.